Hey, boys and girls, weather report for June 1st. Whoa, it's June, and we're having
another month.
April weather in June, but it's like it's going to be like 70 for like a week.
It's crazy, man.
Yeah, it's kind of nice, actually. It's just perfect out there, and it's just warm winds and gentle rains, and I don't know. And then while we get some crazy, like living on the Minnesota River Valley, when I look at the radar, there's these big thunderstorms that come in. They split around the Minnesota River Valley. So I don't think, like just north of us, people get trees down and everything. We get protected from a lot of stuff, which is kind of cool. I wonder if that's a common thing for an east-west running river valley. I don't know. But we've lucked out. Our neighbors haven't so much up north. Had some trees down. And anyway, Donny Dumbo and the fruit bat. I probably shouldn't call Donny that. I don't think he calls me names, but
what inspired you to represent him with Dumbo the elephant?
Because I was thinking Donny Dumbo and the fruit bat, and it just got stuck in my head because I was listening to him, and that's what I came up with.
The odd workings of Speed's mind.
Yeah, it's problematic, actually. So anyway, the first thing I want to do is I made a mistake yesterday when I was talking to Rebecca. I showed this particular diagram, and okay, so the line transposon exists in the same place in both the chimp and the human. But the insertion of the ERV only happened in the chimp. So, and I said that they were in both of them. They aren't. But yeah, but I mean, it's still just as stunning that they're in very similar positions. I guess the flanking genes are the same, and the flanking DNA is the same on the chimp and the human. And so, and then on the chimp, we can see where a line inserted is, or an ERV inserted itself after we split from the chimpanzee and stuff. And let's see, I should probably link that other article. Now, there's an article by a creationist named Anjanette Roberts, who's a molecular biologist, and she works in viruses. And she has a pretty good article, actually, where she levels some, reasonable criticisms, but she takes them way too far. At the end, she kind of sums it up and said, well, she has examining presuppositions, and insertion sites are not random. She goes through that. Important sidebars where she tries to complain that the genome hasn't been completely sequenced. Well, that's probably not so much the case as it was in 2017. But there's a lot of centromeres and things like that we haven't completely sequenced because they're so repetitive. And then she goes on. What did she end up with?
Did you say she went over the insertion site or integration site isn't totally random thing?
Right. Yeah. She
I mean, which is correct. It's not totally random, but it's pretty darn close to random.
Well, she suggests that between the human and the chimp, all of these things that happen in the same spot, showing common ancestry, she is speculating that it inserted at exactly a particular sequence of nucleic acids, right? And that might have happened once or twice, but for the most part, the insertion isn't quite that precise.
Right. It's very unlikely most of the time.
Yeah. and just all sorts of stuff with that. Let's see. And then ERVs and REs. What the hell's an RE? I can't figure out what that is.
An RE. Wait, RV or RE?
RE.
RE. I don't know.
A recording engineer.
Scott.
Race car.
Andrew.
Engineer.
A religious enthusiast.
She never tells you what it is, and I can't really find a lot of references that talk about them. So, okay, and then she says evolutionary presuppositions need to change. Okay, that's what she comes up with after all this, and it's like, no, nobody's making presuppositions. There's at every step of the way, everybody is checking the work pretty thoroughly. So I don't.
Is she making use of buzzwords to kind of appease her creationist wannabe friends?
Yeah, she's pretty much in that camp right now. So anyway, I'll link that article in the description.
Okay.
It was Fuzzrana did an article, and boy, did he do a bad job. Holy shit, he said some crazy shit.
Like what?
Oh, I'd have to pull it up to remember.
Oh. Hey, we got time.
Yeah, I guess we do. Anyway, but I'm doing a update here. It's in fact I linked from a Fuzzrana article to this one, so that's how I got here. And let's see if I can find Fuzzrana's article. Yeah, okay. So he's going on about endogenous retroviruses help fight tumors. And also when we show the SFT video, RawMat and SFT go on about how endogenous retroviruses fight cancer, right?
Right.
And so I went and looked up.
They have functions for fighting these things, yeah. I remember that.
Yeah, like Fuzzrana says, a Swedish lab recently found some sort of function, or so, I forget how he put it. Let's see. He's talking about the P53 protein and how there are ways to activate the P53 protein, which, let's see, how the hell is that? And it ends up activating ERVs instead of shutting them down, which is normally what it does. The P53 protein normally shuts ERVs down, but there's a way to activate them. Anyway, so Fuzzrana and SFT and RawMat are going on about how ERVs fight cancer. No, they don't. If you go look at the Swedish paper that they're pointing to, the paper is about what happens or how you can pharmacologically manipulate the biochemistry of P53 to turn on these ERVs and target cells for removal by our immune system, right? But it's all about pharmacology, and the reason that you get cancer in the first place, they point out in this paper, is oftentimes because of these ERVs. So the ERVs cause cancer, and they're talking about people that become immune to the therapy that they're going through. Basically, therapy like chemotherapy works to a certain level, and then they're talking about ways to manipulate it after that, so they can get these ERVs to turn on and get the immune system to recognize the cells as a virus. So that's what it was about. I can't find anything about ERVs fighting cancer. They don't. They're I think in one paper, I think it said like 50% of all cancers are caused by them. They're not a protection for cancer. It's just ridiculous.
To answer Ian Chen's question from the side chat, no, he did not. He did not address Dr. Dan's actual critique. As far as I can tell, he kind of superficially addressed the kind of minor details of Dr. Dan's review and say, oh, well, he didn't talk about this, he didn't talk about that. And
yeah,
like, well, what Dr. Dan put earlier, he pretended to review it, but he didn't actually address anything he was saying or the main points he was making.
Let's see. There's a paper by Marie Dewanu and Thierry Heitman called Endogenous Retroviruses, Acquisition, Amplification, and Taming of Genome Invaders. I think it is.
Are you trying to share these things?
No, I'm not. I'm just
Okay.
I'm just about to link. I'm about to put the name of this paper. I'm trying to think if this is the cancer paper. Anyway, I'm trying to do like 5 different things at once here. So I'm going to link that paper by title.
I'm assuming you also linked the actual SFT video in the description.
I did, yeah.
Okay.
So anyway, okay, so here's what the fruit bat says. Okay, let me get my stuff together here.
Oh, are you going to share the video already?
Yeah, a little bit of it.
Okay.
Yeah, this is just one part. See how we're talking about cancer. So,
Oh, we're going to go for it piece by piece, I see.
yeah.
Out of the way. Well, what are endogenous retroviruses doing helping fight cancers? Most cancers aren't something that kids have to worry about. Cancers come much later in life. So what's...
Okay.
I guess he's never been to Memphis where they have that hospital for children who are dying
So
of cancer.
So 16,000 kids are diagnosed with cancer in the United States every year. 400,000 worldwide. In the United States, 1,600 kids die every year, and that's 1 to 14. And worldwide, it's 100,000 kids die of cancer every year. And in fact, cancer in the United States is the #1 cause of death in the age group 1 to 14. So I when he said that, and he's got kids of his own that are playing in the background, I'm thinking, what a tool. But anyway,
What's the point of preserving something that's a backup mechanism to preserve you from dying from cancer late in life? That makes absolutely no sense. But before I digress, let's specifically talk about Dan's argument and how it actually hurts himself. And
okay, we're going to have to back up here because they're talking about this silly idea. Anyway, okay. Silly idea.
So,
So,
Matt, this was one.
Okay, so Dr. Dan basically talks about DNA methylation. Most ERVs are shut off by DNA methylation in the human genome. They don't actually, very few of them actually get transcribed.
Okay.
And, yeah, but SFT thinks a lot of them do. ERVs we're talking about. Now, other transposons like lines, that's a little bit different. And that's one of the things that, well, I don't think they understand the difference between the different types. So they're always talking about ERVs, and ERVs are, I think we have, 6,000 or 10,000 of them in our genome, amounts to about 8%, but 50% of our genome are occupied by lines and signs and other kinds of transposons, right? And they never want to talk about them. Yet they do talk about them, because when they talk about transposons that activate genes, they're usually talking about lines. Well, sometimes the ERVs will do it. That long terminal repeat on each end of an ERV, it's this little identical piece of DNA that's plugged into the beginning and the end. Those things have promoters on them. They will attach to promoters. And if they jump into an area right before a gene, they can change the promotion of that gene. And one of two things will happen, or 3 things. Either the organism will die because of that mutation, or nothing will happen because it doesn't have any effect, or it will have some benefit somewhere along the line. So it's the negative, positive mutations, and most of them are neutral. That's right.
And which type of transposon are you talking about here?
Both ERVs and lines do them. Almost any transposon. Well, transposons are actually little pieces of DNA that promote themselves. They have their own promoter at the beginning of them so that they can transcribe if they aren't methylated out of commission, right?
Right.
If the DNA gets is loose enough to transcribe, they have their own transcription factors. And if they insert a head of a gene, okay, that transcription factor can bind and it can transcribe that whole string of genes, basically. So they can upregulate a gene, and they think that was one of the contributing factors to evolution, that these transposons basically were a serious enough mutation that they could change transcription of a set of genes. So basically any of them, because they have their own promoters built into them. Most of them in our genome, though, are so mutated that the promoters probably don't even work anymore, except for the ones that were selected because they did something beneficial.
Right.
And I've looked. I've got a paper here that links. Okay, there's hundreds of them they found that are transcriptionally active like that. They will activate a gene, that they have some regulatory effect on gene. Hundreds of them out of millions. And as far as functional transposons, I think I found 27 in the human, something like that we've identified, that have some function. And they found them in fruit flies, and they found them in other mammals and stuff. So they're all over the place. So I mean, it's a little of this and a little of that, but I mean, 27 out of 3 million transposons in the human genome. Come on, you telling me that the writing's on the wall there?
Yeah,
that does not overturn junk DNA. So sorry. Anyway, okay.
So
Matt, this was one specific comment that I found rather fascinating. Clearly, a lot of the critics, they still don't understand the fact that if the majority of our genome is not the result of deep time evolution, okay? If the majority of the genome does not reflect common descent, which means that, again, the majority of the genome should consist of what? Evolutionary leftovers, genomic fossils, and junk DNA, okay?
No.
If that's not the case, and what we actually see is a genome of function, okay, in a nutshell,
there's no reason for high functionality in the genome to overturn evolution. What it would overturn is biochemistry, because we wouldn't have any room for mutation somatically, right? If everything was like functional and had to be just perfect and pristine, we have mutations all the time in our somatic line. Life couldn't happen. So it's life that couldn't happen without some junk DNA. It's not evolution. Evolution doesn't matter. Are you talking to your dog, Scott?
Oh, where'd he go?
I'm muted.
Ah.
So any talking you hear is
No,
either in your head or out the door.
No, it's like I hear something like, the fuck is that? I don't know. It's like,
Oh, well.
My dog might be snoring in the background. Is that what
oh
you're
yeah,
hearing?
your dog is snoring. That's it. Okay.
Oh, sorry.
Okay.
I guess I'll mute myself.
the book goes into this in great detail. So for sake of time, I'm just going to say, in a nutshell, what that means is what we're looking at is patterns, nested hierarchical patterns by design and not descent. Okay, that's the differentiating line of evidence. One differentiating line of evidence, that is, when it comes to...
Okay, hold on for a second. Or mannequins or whatever. Did he say by design and not descent?
Yeah, the two.
That's a stupid.
The two different theories. And so he's basically saying, well, it's either design or it's descent with modification.
But organisms still descend from other organisms even if they're designed.
Yeah, well, that too.
That is true. I will say,
False dichotomy much?
yeah.
Well, he's, I don't know. Let's go on.
Descent is function? Is the majority of the genome functional, or is the majority of the genome junk? If junk,
The majority.
Or and just consider this, SFT. Maybe you don't understand the evidence for common descent as well as you think you do.
Right.
Just
because-
throwing that out there. Just pinning that. Okay.
because
Thanks.
it isn't just functionality, it's those mutations, those differences between the genomes that we get the phylogenies from, right? We don't get it from common function. I think he thinks that we're putting it together like his car, his car idea, like
Yeah.
he would like it's statistics.
Like we're just looking at the superficial outline of the organism and saying, oh, it looks similar, therefore it's related or whatever.
Yeah,
Or it's more closely related, like a sedan and a Honda or Chevy, whatnot. I don't know many cars.
Erica scientifically calls that eyeballing it.
Yeah.
Yes,
So,
that's the term I was looking for.
He thinks
not doing
we're putting together traits, like different traits. Like, okay, the frog has an enzyme that does this, and we have an enzyme that does that. It does the same thing, right? So basically, and then we go back further, before a frog, and we don't find that enzyme, and then we're saying, oh, we have more in common with the frog than or whatever. Okay. Well, we have more in common with a chicken than we do with like a starfish, right? That sort of thing. He thinks we're doing it by traits. He doesn't understand that it's also done statistically by mutations, because that enzyme in the frog and the enzyme in the human, while they do the same thing and they have roughly the same protein structure, they have a lot of mutations in that gene that we can look at, and that's the stuff that we're looking at.
Right. Yeah, and that's an odd thing for a designer to do. Put these proteins with about the same basic function, like especially for biochemical purposes, and put a bunch of these mostly silent mutations in them all over the place. Oh, what? Why would you do that? Like it doesn't make sense from a design perspective.
Or swap out one polar amino acid for another, or one non-polar for another, right? Why would he do that?
Right.
That's
the most reasonable expectation from a design perspective would be he either makes it the exact same sequence or roughly the same sequence, or he makes it very different sequence. He shouldn't necessarily a priori make it this sequence that follows a nested hierarchy, despite the fact that it doesn't need to. It's just the logic.
Yep, logic.
It gets me.
Where are we? 7:51. So I've gone past function. Okay, so he's talking about function. Yeah.
junk DNA is overturned.
Oh yeah.
Then with that overturns endogenous retroviruses
No.
for common descent. It also
So
Okay. I was. Yeah, go ahead. Sorry.
He doesn't get it, right? He doesn't know how to do it, right?
Well, I was pointing this out to me pointing this out to him in the comment that went away for some reason the other day, that even if all these ERVs had function, that still doesn't explain the fact that they are located in the exact same position on the chromosome. Because if you look at any particular gene, it doesn't matter where that gene is located for it to have the particular function that it does, or to perform that function in an efficient way, basically. So God, the designer, could have had these exact same ERVs, maybe with the same mutations, and he could have placed them all around the genome, and they still would have functioned the same way. He didn't need to place them at the same sites in these different species, accounting for chromosomal rearrangements and all of that, of course. So even with the function, which doesn't itself work, even assuming that they do have function, his argument still doesn't work. So, among other things.
Yep.
So
Okay. Oh, let's see. Do I want to divert? No, not yet. We'll do it later.
So it gets into a whole nother field, a whole nother topic with
we
mutation
go.
accumulation.
I
Okay.
think that's it.
Even if the genome is only 10% functional, that's too much, given what we know about low impact, nearly neutral, and deleterious mutation accumulation. If the genome is 50% or more functional, then evolution can't happen. That's it.
If the genome is 50% or more function, then life can't happen. I don't think any of us would live past 2 or 3 months old. I don't know. There has to be a lot of flex room, and because of copying is not perfect, and our cells divide all of the time. It just wouldn't work, and it has nothing to do with evolution. They used to, well, in fact, I was reading a paper or a book somewhere just yesterday, and they actually thought everything was functional back before like the 1980s, when the selfish gene thing came up, and they were surprised that everything wasn't functional. So when they discovered all these sequences, they thought, well, we're going to find function for them, and then they kind of went the other way, and the tides turned, and then they were down to like, yeah, it's all junk, and then the tides turned back again, and now it just goes back and forth. Anyway.
Evolution's done. Okay. But anyways, Matt, what I want to do, brother, is just go over this comment. I found this one funny. Again, this is just a teaser. There's just a couple comments here and there that we're just too excited. We just kind of want to respond to and give some thoughts, right? Before we do a very technical video series response, as well as I've pointed out, a mini handbook for people to check out, the Endogenous Retroviruses.
I think he gets to it here.
He put forth this comment, this argument, to the fact that the majority of the genome has evidence for activity, okay, biochemical activity, which suggests function, which is why biblical creationists have made testable predictions, testable and falsifiable predictions on.
What the fuck are these predictions they make? When have they ever made a prediction?
we have predictions. Well, show me what the predictions are.
They're going back and they're claiming, he's claiming that they predicted it's all going to be functional.
Oh yeah, retrodictions.
And now we found out it's all functional. No, we haven't. We're still running at around 8% functionality in the damn genome. That's all we know about. So I mean, it isn't. I don't know where he gets his prediction stuff from. Testable predictions. What the hell is he talking about? I wish he'd like lay one of them out for us.
Yeah, and who's doing the testing?
I have a prediction. And we're going to test it. Yeah, exactly. Anyway.
I mean, even if much of the genome has some minor function, for example, that doesn't necessarily mean that all of those sequences matter necessarily to the organism. That's kind of the crucial difference there. Any kind of DNA sequence can have some function, but that doesn't mean that the organism depends life or death on that particular sequence. Yeah. And we know that much of the genome is like that. The organism can do just fine without it.
Yeah. Okay, I think he gets to the energy thing.
Including function in endogenous retroviruses, loose sequences, pseudogenes, all kinds of genetic sequences. And so the argument goes from the critics, what we're looking at, the majority of this activity is spurious, or it's nothing more than genetic noise, right? And of course, in the book, I absolutely demolish that in great detail. This video is meant to be shorter, so we're not going to go into all the details. That's what the book's for. We're just kind of showing that this specific critic, and I do appreciate his feedback, and the ripple effect of that is ongoing discussion pertaining to this important topic. But the responses to that argument were essentially ignored and dodged. Anyways, so Christian Myth says here, and then this spectacular lack of insight Okay, and he quotes a portion of my book where he says there should be evolutionary processes that would end up suppressing this transcription in an effort to preserve energy and resources. So he says, you think? Okay, and for more context, it's because as biblical creationists, Matt, as you know, we would suggest, okay, the majority of this activity, the reason why there is so much activity is because it's beneficial to the organism. It's doing something. It's there for a reason, right? And so we can make testable predictions because we like to point out the trajectory of discovery favors genome-wide functionality.
One of the papers that he shows in this video somewhere, I was reading it just before we went live, and it says that most of the ERVs are methylated and very few of them actually transcribe, except under extreme stress or shock. But then that gets to Matt's firefighter analogy later on. But so they, so actually there is in fact, some people have hypothesized that the reason that epigenetics evolved was to suppress transposons. That was actually, that's where methylation came into favor, because it massively depressed them. But then the other thing, they're going on about energy, and I think we may have skid by the part, but they said, you know, evolution would have gotten rid of those things that it's not using. If you don't use it, you know,
not necessarily.
No, not at all. Because for one thing, eukaryotic cells produce a lot of energy, waste a ton of energy. There's a lot of proteins that are just ripped apart. There's a lot of mistakes made, and there's a lot of energy left over. So no, and evolution doesn't sit and think about, you know, its efficiency. Right.
Well, much of evolution is also neutral. It's not necessarily selection just acting on it.
Yeah, I mean, if the cell survives, evolution is fine, and that's all natural selection does. It doesn't sit there and write to the Congress and say, you're spending too much energy on these ERVs. We have to get rid of them.
Kind of like the misconception, survival of the fittest. It's more like survival of the fit, or the good enough, basically. If it gets the job done, then it's going to most likely survive and reproduce. That's the point here. Right. Same thing with this.
Yep. Okay.
And if the majority of this really is genetic noise or spurious, right, then for one, evolutionary processes over deep time should have weeded out a lot of this genetic baggage. I mean, it's very wasteful of energy and resources.
And besides that, these things were actively jumping at, you know, some point after they initially entered our genome. They were active for possibly billions of years, and that's why we have so damn many copies. So, I mean, they were actively jumping. If evolution isn't going to corner every one of them and cut them out, I mean, Jesus Christ. See, he's thinking in terms of the creator. You know, he's thinking in design. He's thinking teleologically, and that's,
he seems to be getting it like twisted around himself too, because he's saying evolution predicts junk DNA, but oh wait, evolution should have gotten rid of junk DNA. So I mean, like, which is it? Which you're going for here?
Yeah, well, it's in his mind. I'm sure it's a big jumble, and it ends up with pictures of Jesus at the end of every jumble. So
yeah, oh yeah, those are some good videos, Chris. Yeah.
Which one? Oh, John Perry did it.
Yeah. Where he responds to SFT on the ERVs thing.
Oh, cool. God, I wish I had a link for that.
To keep this around and to transcribe a large portion of it. So it's a waste if it's not really doing anything. And there should be mechanisms in the genome that would...
Okay. So anyway, there's that. I think he goes on about that for a while. And then
okay.
And you're back. You're back, and you're ready to destroy some evolutionary rescue devices.
All right. Well, the first thing that came to my mind was evolution says if you don't use it, you lose it. That's a term that we hear today all the time, right? Well, that would make sense. The body's not going to be wasting energy and supplying it to something that's going to be worthless and deemed not necessary. Remember, evolution only wants to get you to the point where you can reproduce and then get out of the way. Well, what are endogenous retroviruses doing, helping fight cancers?
Wait,
Most
wait,
cancers.
go back a few seconds.
Okay.
Produce and then get out of the way. Well, what are endogenous point where you can, not necessary.
Yeah.
Remember, evolution only wants to get you to the point where you can reproduce and then get out of the way.
Okay,
Evolution.
he just explained it.
No.
He
Evolution.
just explained why.
Yeah.
Oh my goodness. Matt.
Okay, so then let's see. He goes on about cancer for a minute here.
Well, what aren't something that kids have to worry about? Something that's a backup mechanism to preserve you from dying from cancer late in life. That makes absolutely no sense. But before I digress, let's specifically talk about Dan's argument and how it actually hurts himself. And I'll tell you why. He is exposing that junk DNA is not really actually being junk, but actually involved by methylation factors, meaning this is an epigenetic signature. This is what's going on to flip genes activations on and off. And I like to think of ERVs like genes that...
Okay, so we got to listen to that again, okay? Because listen to how confused this guy's mind is.
Being junk, but actually involved by methylation factors, meaning this is an epigenetic signature. This is what's going on.
What the fuck is he
to
talking
flip
about?
genes activations on and off. And I like to think of ERVs like genes that work in different stages. They aren't fully operational 24/7. They're not a workforce that's always working perpetually. Rather, they're like a crew of firefighters that are ready for the alarm to go off and get to work at any time.
Yeah, you get this whole firefighter thing that he goes on and on about. That, you know, the crew of firefighters, and
It seems like the whole thing when they say, or when they talk about the genetic code and how it's redundant. Oh, well, sometimes you need a spare tire to get across in your car in case one breaks down. But
yeah, and here's the rub. Sometimes you need something in your trunk because you may get into a environment where what you had before no longer works. And so now you bring out that neutral DNA, and that neutral DNA now has a new function.
But you don't want too much junk in the trunk.
And that is what evolution is, you
right, that's how co-option works.
Or can there really be too much junk in the trunk? There's a debate there.
Oh, yeah.
It's just amazing how much they're not thinking through this hard enough.
But that's
Some people
they're not
really
thinking
like
about what they're saying.
junk in the trunk.
The irony is that they're describing evolution, right?
Right.
How it works. And they're saying all this actually, all
Oh,
this stuff.
but that's not evolution.
No, that's not evolution. Because it was already in the trunk, right?
Right.
Yeah. Fuck. Anyway.
So I want you to think about it. We have some that work for placental development. We have one that specifically works in stage 2 placental development. We have others that...
We have one little piece of ERV that turned the envelope protein that ended up having something to do with placental animals.
Right. So not a complete ERV, one of the genes in an ERV. Like you said, it's the ENV gene, right?
One on the end.
Yeah.
Yeah.
Which makes sense because that's what viruses use to fool the cells in the first place through those glycoproteins to get in and invade the cell to begin with.
And they go on about later on. I'm not sure if we're going to have the patience to go through all this, but they go on later on about certain ERV, certain transposons will trigger an immune reaction. Basically, if they get on the coat of the cells or whatever, they'll trigger an immune reaction. And he says that's why they have to be a lot like viruses, and that's why God made them the same as viruses. They have to trigger the immune reaction. But did God need to put 3 million of them in there?
Right.
One would have been enough. Just
right. Maybe a handful, but why do you do it so many times in the same place? It doesn't work. It's too contrived. It doesn't explain anything. Or
And
I
that's
should
just-
say it explains some things, but it doesn't explain it nearly as well as a common descent kind of explanation.
Yeah, it doesn't explain
That's
why,
far more the parsimonious one.
and it doesn't explain why they're still jumping around actively inside the genome too.
Right.
I mean,
Yeah.
if they were just there for defense, they wouldn't have had the
The machinery to copy and paste themselves, basically.
yeah.
A quick diversion here. Andrew, have you been fighting a cold lately?
No.
Your voice sounds deeper.
Well,
At first, I
am
was
getting
thinking, well,
older.
you had a cold, but I think your voice is getting deeper.
Yeah, I think so too. Is that a good or a bad thing here?
Our teen scientist is growing up.
Oh yeah.
Oh, thanks.
Anyway, God should have left out that reverse transcriptase, because then it
Yeah.
wouldn't have jumped around the gene, right? Then it could have just went out on the surface of the cell and
Yeah. What's the purpose of putting a reverse transcriptase next to a different gene that has a function like? I mean, maybe you could repurpose the POL gene by itself to make reverse transcriptase and co-opt it for something else, but that's only like we were saying before. It happens to co-opt certain genes within a ERV, but the whole ERV itself doesn't do a brand new function.
And why did God make viruses again? Okay, so he made viruses because Adam ate the apple.
Right.
Yet he put all these ERVs in Adam.
He put all these viruses in us to fight against viruses.
I guess so, yeah.
That seems a little extreme and inefficient, don't you think?
Yeah, I don't know. That story just doesn't make any sense. Junk DNA overturned. Oh my goodness. Oh, where am I at here? Okay, I'm at. Oh my god.
Where are we at? We are on Earth.
Let's see. Crew of firefighters. So we have crew of firefighters by Ramat. And I don't think we need to listen to his stupid analogy. Let's see. And then we have listening has a
to go off and get to work at any time. So I want you to think about it. We have some that work for placental development. We have one that specifically works in stage 2 placental development. We have others that work to fight viruses and infections. We have others that fight cancer, like I mentioned earlier. Well, all of these things are individualized specific functions that work in stages. And if that's true, then think about it. Do you have a virus attacking you right now? Probably not. But somebody listening right now actually does. And
somebody has a virus attacking them right now. And guess what? God has just the ERV for you. Anyway, okay. And Donny,
Except he didn't have the ERV for the people who died of COVID or of
right.
or who are afflicted with malaria. But malaria is malaria a virus, or is it caused by bacteria?
It's a protist, I believe.
Oh, that's a whole different thing, huh?
Yeah, I think I don't think it's a bacteria or a virus, bacterium or virus. I was gonna say it's kind of like that one woman. Here, you get an ERV. You get an ERV. Everyone gets an ERV.
Yeah,
you're referring to Oprah Winfrey.
Yeah, there we go. Okay, I don't know why I couldn't remember her name.
I've caught a lot of interesting things out in my yard, but I've never caught malaria. So I'll have to lay a trap.
So according to Dan, these are
here we go. Unexpected.
Yeah, notice this. We have paper after paper. This is a couple here. Recent studies also point towards and until we have paper after paper. We have paper after paper. We have paper after paper. This is a couple here. Recent studies also point towards and until now unexpected. But no, Dan says it's expected, and I've already challenged.
So he's going on because in this paper they said it was unexpected. Something recent studies also point to an until now unexpected role of the immune system for the control of these elements. Okay, so
hey, is that the abstract of the paper?
Yeah, but the paper that he's talking about, that's that Duano paper that I have up in front of me. And the whole paper is about how the immune system has been sort of commandeered to get rid of ERVs to keep them down, because sometimes, because they're up, and I didn't know this, but there's a method of transposition where they actually create something that goes out to the outside of the cell and then comes back in, reinvades the cell, that sort of thing. It's like almost functioning endogenous retrovirus. At least that's what this paper shows, and I've linked that paper in the description. But anyway, the unexpected thing is that they found that the immune system can be triggered by these things, and I don't know what the hell he's talking about. But whether or not, whether or not some particular biologist expects something or not, or is surprised by something, that happens all the time, right?
Right.
I mean, everybody has different opinions about what we're going to find and stuff like. And he goes on and on about this for about 5 minutes. He doesn't like the unexpected thing.
Well, like I think a good way of phrasing it is there are probably a lot of things that scientists didn't expect before, but once they took a fresh look at it, basically, and kind of found a way to say, oh, well, this actually does make sense in light of what we now know and what we should have expected, given the basis or the foundations of our idea. So, like you said, it happens all the time. It's not as big a deal as he's making it out to be.
And they've been told over and over again not to take the abstract seriously when people use these exclamations and they say, oh, we were surprised that, or when they state the problem, as Dr. Dan tried to point out to them, that a lot of times in the abstract, they will state the problem. That's not the conclusion of the paper. That's the problem the paper is going to solve, right?
It's like taking the hook out of an essay and saying that's the conclusion of the essay.
Yeah, he's basically got this paper that I've, let's see, I can show it here. Logicam, PPT. Oh, I got to go down there. PDFs. Okay, this one. So he's got this paper and he's showing the abstract basically, and he's got this part highlighted, and that's all he ever clips. That's all he ever looks at. Is the abstract. But if you go on and read this paper, it's about a whole lot of other stuff that. So I don't even know why he would bring this paper up. It doesn't make any goddamn sense to me at all. So let's see.
So you're saying he brought up a mostly irrelevant topic?
Yeah, because it said unexpected.
That little word.
Yeah. And somehow it goes to his functionality thing because he thinks somehow they're functional. When actually this paper is about how the body fights against these goddamn things. Yeah.
It's so funny how they're so functional, yet so many of them cause so many diseases when they are functional.
Yep. Anyway, let's see what else we got here. Oh, so I went and took apart a squirrel monkey retrovirus, and let's see if I can move it over a little bit. Yep, I can move it over a little. I moved it over too much. Let me get rid of me. It's not about me. It's not me.
So is that what your family pictures look like on the mantle and on the wall?
Right.
Just hang all these T's and C's and G's, etc.
That'd be actually kind of cool, though. I might steal the idea.
I'm a little surprised that you can't see this whole. Oh, there it is. What the hell? Okay. There's the first LTR. You see TG, TT, GGG, and then way down here, this is the guts of the virus right here. And then I found the envelope, the part that codes the envelope protein.
I hate those gut viruses.
Yep. And at the end of that is the, let's see, the second LTR, except where did I put the thing for it? Oh my goodness.
LTR2?
Yeah, LTR. Oh, do you see it? Yeah, there it is. Oh, LTR2, yeah. Okay, you can see it. I just can't see it for some reason. Anyway, so I went down, and then I took an actual ERV, which is from chromosome 1, and this is let's see, I think this is ERV K9, and I could not find any homology at all. This is just a bunch of garbage. It's a bunch of mutated garbage. And over here, I have, let's see here. Oh boy, why is that? Okay. Over here, I have a list of, I think, let's see, 115. I forget how many I have. I wish I had positioned my screen a little more accurately, but I haven't. Let's go down the end here. Well, these are all the ERVs in the human genome. Basically, some of them. You see, some of them are really, truncated and tiny, and others are, bigger, and it just goes on and on and on. And let's see.
That's kind of weird for the designer to do. Put those.
It's weird for the designer to do that.
Little tiny fragments here and there, and then big ones.
I think in this file there are 3,690,000. That can't be. So they found 3,690,000. I think they're numbering them sequentially. Maybe they skipped around. 108 to 1. Yeah, these are sequential. So this is a pretty big file. I mean, it takes a while for this file to load, and some of them are gigantic. Look at that.
Oh wow,
that's a huge one. Anyway, so there's a database called RetBase that has all of the, I think it has all the signs and the lines too and stuff, but I've only downloaded the CRV one.
How many gigabytes did that cost you?
It was in a tar file, so let's see. I don't know. I'd have to look. Not sure. I'm sure it's quite a bit.
Yeah.
Might run around a gig or two, something like that. I downloaded worse, but yeah. And then I was like screwing around with different ones and trying to figure out various things. But I'm trying to find really good examples where I can look at the flanking genes. And I think one of the ones I'm going to go after is this lady that I mentioned before, that Roberts, Dr. Roberts. She came up with a gene that basically it's more mutated in chimpanzees than it is in humans, even though it's in the same spot. So she was suggesting that, the designer did it. And, our problem is that why does it look like it was inserted?
Right. Well, why aren't all the other ones in the chimp genome nearly as mutated then?
Yeah. And a lot of them maybe, we don't know, and it could be a high mutation rate. It could be that something weird happened. I don't know. We'd have to look into it. I would look at the flanking area and find out if that was a particular hotspot for mutations or whatever.
Yeah.
And because he goes on about the Y chromosome of chimpanzees all the time in primates. Well, the Y chromosome mutates like crazy. I mean, you go from, in fact, within a species, I think you can find amazing differences in the Y chromosome. And with chimpanzees, the Y chromosome has diverged from ours tremendously.
And just so you're not quoted out of context here, because I know you will be, what you're saying here is not that there are mutational hotspots that cause the specific shared little point mutations. You're just showing an example where it might be a mutational hotspot that just caused, on average, more mutations in that general area.
Yeah, you'd have to look at the type of mutations and exactly what happened. I mean, it could have been, been some weird duplicate.
I'm just throwing that out there because I know some SFT followers probably going to misquote you there.
They probably will.
Because he does like that mutational hotspot idea.
Yep. We got some fruit bat thoughts here at the end.
Any thoughts, brother?
He won't be able to find it. Matter of fact, if you go back to the early days of endogenous retroviruses, you don't find anybody making predictions because they immediately assumed evolution is true. Therefore, they have no function at all. They're genome.
And that isn't what they were thinking in the early days at all. He talks about Barbara McClintock, and what she came up with, her idea for that there were jumping genes. Okay, they said no way, basically, and they ignored her for about 10 years, and then they gave her the Nobel Prize. But by that time, they were saying that all of this stuff has to have some function, right? So I mean, like I say, it was every individual scientist is going to have his own opinion about this shit.
Right.
And he's going on about that. I don't know.
We'll make mummies. There's
I'm not showing the video, am I?
Nope.
Well, we'll have to fix that.
Rust hot
zombies.
spot.
That's all that's left. It's just a shell.
Wait
There's
a minute.
another genomic.
Did you see that?
Mummies.
Rust hotspots on vehicles? What the hell is he talking
Oh, I bet that's the mutational hotspot thing again.
about? There it is, yeah.
There's zombies. That's all that's left. It's just a shell. There's nothing. Matter of fact, I hunted it down, and I found the first person to ever make a prediction that they might have a function. Not what type they would have, but that they actually do. Her name was Barbara Mick.
He can't even say it. It's like, Jesus Christ.
Barbara
he
McBarbara.
He just now hunted down Barbara McClintock.
Clintock.
Like last
She was
week.
born in 1902, and she made a prediction in 1975 that ERVs might have some type of a function. And
Not really, no. She did, basically, she found out that it had an effect on the color of maize on corn ears, right?
Yeah. Well, those weren't even ERVs. Those were just transposons.
They were, I forget what kind of PLEs or something like that.
Yeah.
I forget what their name. But anyway, she had this theory that they were jumping, and they were jumping in the genome, and that's the part that nobody believed, right?
Right.
She did not predict that they were functional. She was she was showing how they changed the color from purple to yellow on these maize ears that she had, based on there's like some polyploid crazy genome, and as it goes from one corn kernel to the other, a transposon will change the regulation of that color, and they'll turn it from purple to yellow or yellow to purple. And I think it was about that. So anyway, that was the whole Barbara McClintock thing. But they got it. Of course, they're going to get that wrong, because it's all they really want to do is cherry pick, right?
Yeah.
Does he have any other fruit?
and they just
thoughts?
laughed at her. It was just ruled out as an idea. Didn't even get into it for decades later. Decades later did they even
I
consider
think she was
what
highly
talking
respected.
about. So she was the first one. She won a Nobel Prize in 1983 for in medicine or something.
No,
But
no,
I was
she
just
won a
looking at
Nobel
what
Prize
her
for. I think she won a Nobel Prize for her work on corn maize, right? It wasn't in medicine that I know of.
concepts were on endogenous retroviruses, because I wanted to figure out when was the first person to actually say anything about this possible function. And no, they were not predicting function. They predicted dead, worthless.
Again, it doesn't matter either way.
She wasn't predicting function. She was showing how they mutate
Right.
basically the genome. She was showing a mutation that had a phenotypic effect. That's what she was showing. Anyway, fuck these guys. I'm done.
You're done.
I'm done with these guys for now.
Well, I think we've pretty accurately shown their thinking process here.
Yeah.
At least for some part of their video response.
Yeah, there's more of it. I like I say, I'm at 46, and I'll see if I can glean any more.
Yeah.
Of it.
Who knows, viewers? We might have a part 2. We'll see.
They're going to come back and say, "Oh,
There's always going to be more time for ERVs and genes."
Yeah.
I mean, like actually learning about this stuff, it takes your breath away, and I don't understand why people go out of their way to misrepresent the science about this and about
Because
evolution
of
in
the
general.
intervening religious presuppositions. And
Yeah.
references. That's all it is. It's twisting, their view of reality. They're shaping their view of things based on this insistence that the Bible be literally true.
Yeah. Oh, yeah, you might be right there, Benghazi. I'm sorry.
Oh, let's see here. What do I got? Can I show this?
Well, they probably are thinking. They're just not thinking through it clearly enough. not thinking about the consequences of what they're saying.
Well, they're okay. There's one part that I should play, and I think I can just play the audio.
Okay.
We got Danny.
Yes, by all means.
Let's see. Oh God, I didn't.
Everybody looking forward to that Dr. Dan thing. That's tonight, right? At 9 or 8?
Yeah.
Yeah,
it's 6 Pacific Standard Time, so it'd be 8 Central and
8 Central,
9 Eastern. Yeah.
Exactly.
Yeah,
That'll be
I'm
good.
looking forward to it. That'll be fun to watch.
Oh, that's tonight. That's right. Cool.
Yeah.
Yeah.
So
Erica
we'll see what goes.
just completed a premiere where she does a review, continues her review of contested bones.
Yeah.
Oh yeah.
Talking about Homo erectus, which I think I need to find some, artist renditions of what Homo erectus actually looked like, based on what we know.
Yeah. Homo erectus via a 2022 rendition, basically.
Mm-hmm. Yeah, and they were highly successful, weren't they?
Oh yeah, they are probably
years.
one of the most successful hominins to ever live.
Yeah.
Yeah,
So
a million years, give or take, I think.
yeah, we could learn some lessons from them. In fact, I'm going to talk to a plastic surgeon and just have everything changed to be Homo erectus like.
Yes.
Fair enough.
So this is. Let's see here.
What is this?
This is the list of ERV papers that I'm looking at.
Ah.
This is my modest collection of papers on transposons. Okay.
Okay.
Yeah. What do I got there? I got to look at info. Get info. Get info. What's info? Let's see. How many items? Where are they? Kind on disk. I cannot see how many items. I don't know what. 268 papers on transposons.
Is Dessel having a debate or something? What's going on in the side chat?
Yes, I know you know him through his other activities, but he's debating Jordan Kareem from Reason to something, or
Reason about.
And on the historicity of Jesus. So they're two very smart gentlemen who do their research are going to debate it on Amy Newman's channel.
Did we get a link to that?
Well, it's not till June 24th. I don't know if it's set up.
Oh, okay.
We'll get to it. We'll get to it. Okay. Anyway, I think that's about all I had today, other than let's see, I had that. Okay. So I was talking to Rebecca yesterday, and she brought up this Minute Earth video, and I linked it in our description. Are we really 99% chimp? And anyway, the video goes through and explains how they align genomes. And like, okay, if we're trying to figure out how similar we are to mice, for instance, right? If we went back to mice, they have a drastically different number of chromosomes than we do. In fact, mice have drastically different numbers of chromosome than other mice do. So
Right.
even if you were to compare a German mouse to an English mouse, you would have the problem that you can't just take what we call chromosome 5 and lay it out on chromosome 5 when one of them has 64 chromosomes and the other one has 28, right?
Right.
And they're both mice, right? Because mice have massive varying numbers of chromosomes. So what you have to do is you have to find the genes and you have to align the areas that you can align. And that's what we do. So we take the aligned areas, and that amounts to, I don't know, we left out, 16% of the repeats? And the other thing is the centromeres on different species differ radically. And I don't know if they've gotten to the bottom of that exactly, but a lot of different species have very different centromeres. So you can't just, so you don't want to compare these repeats, the massive repeats. You want to account for duplications, insertions, chromosome joining, chromosome splitting, chromosome duplication. So it becomes, I mean, you can't just use this naive method that the creationists used and came up with 88%.
Right.
But even if you did, okay, if you use it on other species, you'd find that we were still more similar to the chimp than any other species,
Right.
no matter what method you used.
Right.
So,
And the 90 or so percent number is, or 99, 98% number is for protein coding genes. So
right.
it's good to be specific about what type of genomic region you're talking about.
Yeah.
Especially when you're accounting for models of molecular evolution.
Yeah, and they explain basically what they're doing and how they align the parts that are alignable, you know, and the rest of it is, and the other thing is that it doesn't make sense to align a lot of the junk DNA. Junk DNA.
Yeah.
See, I said it.
Well, I think another thing they talk about, because I know what video you're talking about here, is like if there's a duplication and there's nothing else going on, should that count as one change or just a whole series of changes that were just duplicated? So that's how the thing going on here is okay, how exactly are we counting this? What regions are we looking at? It's a lot more complicated than people think it is the point.
So Rebecca watched that, and what she got out of it is that we're nothing like chimps.
Right.
So,
yeah. Which isn't their point.
Kind of motivated reasoning.
Motivated reasoning. Yeah. Because
Girl wants to hop in here in a minute. So, but now he's got to set up equipment. So,
girl, we're not going to be around that long.
The room.
We're getting the hell out of here.
I need to find a trombone riff. To keep
I think we're gonna shoot her in the head, and I'm gonna go for another 2.5 mile walk.
You're just wanting to get outdoors.
It's so beautiful out. It's just wonderful. I went down by my stream and meditated today.
So do you have to run up to your son with a leash in your mouth and jump up and down?
Yep. And then he puts me in the back of the Tesla, and puts me in the. Okay, the Tesla has a trunk. It also has a frunk.
A front trunk and a back trunk?
A frunk.
A frunk.
Yeah, if you look, okay, they give you a little diagram of the car. They have a touch screen. And if you touch the frunk opens in the front, and then the trunk opens in the back.
Does it do like some sort of, infrared scan of your trunk or your front to make sure nobody's in there when you get in the car at night?
You should have that.
It ought to. I mean, somebody hiding in your trunk.
Yeah. It is some really
in it though without keys.
Yeah, and then when my son leaves me in the car, because he doesn't want to take me into the store because he doesn't want to put a leash on me and haul me around the store. So
that's right.
I can watch Netflix or play games on his Tesla screen.
Because anyway, all the shoppers would want to stop and pet you, and
I can watch SFT's channel in the Tesla. It's got YouTube too.
Wow.
Yeah. All right.
Well. Look at here.
We're shooting it in the head.
What's that? Hold on.
That's really rude. Now that I'm here, the king of atheism is here, and you want to shoot her in the head?
You want to make a channel where we yell at James, and you're like, okay.
All right.
I don't see
Welcome
the correlation.
Romania.
I don't see the correlation with that song in me.
Just listen to the lyrics and think in terms of what I'm feeling when you come in.
Well, so now that the stream can start, what are we talking about today?
You picked the topic, Gromania.
Me. Yes. Yes.
You have 3 minutes to say what you're going to say, and we're shooting it in the head.
Okay. Everybody go subscribe to my channel, Germania, and go subscribe to my other channel, Gerstreams.
I know is what you were going to say.
There you go. Little self-promotion there.
Hey, so Donny had a debate last night between Snake was right, and last night or the night before, and another guy, right?
The transitional fossils.
Right. So if you go to his channel, YouTube said his channel has been removed for some atrocity, something he did.
Oh.
You mean we had an atrocious debater last night?
We had an atrocious debater.
Wait. Whose channel got removed, or was it just a stream?
It was the whole channel got removed. Let's see, Standing for Truth.
SFT.
Oh God, they're standing for. Okay, let me go find it.
Let me look this up.
You gotta show what's going on here.
Wait a minute, where the hell? Okay, transitional fossils was the debate, right?
Yeah.
So okay, if you go to that guy's channel, what the hell is more? His name is Christian. Stop it. Okay, Christopher Sylvius is his name.
Yeah.
And you go to his channel, this account has been terminated for violating YouTube's community guidelines.
Oh.
Yeah.
Oh, so it's not SFT's channel that got removed.
Did you listen to that debate? Yeah, I was going to text you, Andrew, to see if you were listening. That guy was really ignorant. I mean, holy shit, stuff.
Yeah, Stupid Horror Energy was in the side chat kind of making a comment about that, said he's worse than Kent Hovind.
He was.
Oh.
And he actually referenced Kent Hovind in one of his slides.
Oh, no.
As an authority, and it's like, oh, he was really
I think
bad.
I actually talked to him once. Privately.
Hey, did you listen to that Pine Creek short that I sent you where he ended up asking Otangelo, do we need to ground our morals?
Oh, no, I never did listen to it.
Oh, yeah. You ought to pull that up. That's pretty interesting. We can get Gurr to comment on it.
Oh, what is it about?
It's just a
What'd you
short
say?
snippet from a longer Pine Creek live stream where Doug asked the question, do we need to ground our morals? And the person with the last word that he focused in on was Otangelo.
Oh, yeah. That sounds like fun.
Okay, SFT put the wrong link up, so that was it.
Oh, young Earth creationism and thinking hard. Oof.
And why wouldn't he fix it after he figured out that he put the wrong link up? I mean, it was his guest, right? He should have at least plugged his channel. I'll go try to find his channel.
Hey, Speed.
We're leaving. What?
I'm sharing the screen.
You're doing what?
I'm sharing the screen.
Are you?
That's the channel everyone should go to.
All right, I'm removing it.
Oh, we're gonna do better than that. We're gonna. God, what a fucking. Who let him in?
This is some bullshit.
did.
I'm calling the police.
The Fought Police.
This is a violation of your First Amendment rights. You should audit us.
That's right.
You should go straight up Karen on us.
You know, I will never understand how so many conservatives get the whole freedom of speech issue so backwards.
So ass backwards, yeah.
It's like, okay, if YouTube removes a video, or if Twitter or Facebook deletes your post or whatever, that's not a violation of your First Amendment rights.
Right.
It's
Yeah, you
just
have the
simply
right
not.
to. Well, there are certain laws in place, but you have the right to. If you just don't like their voice or something, you can reject them. You know, they don't have a right to be on your platform.
This is the thing that really irritates me. Conservatives, generally speaking, will say, yeah, if a business owner wants to decline service to a person because they're gay, they have that right. But as soon as Twitter kicked off Trump, they said, oh, that's a violation. It's like, no. Going by your same logic, it's a private company. They can, render service or not to whoever they want based on their own logic. So, yeah.
Yeah, well, when it comes to making cakes, they want them to have rights. So,
Andrew, what is up with your name right now?
Because I know you'd see it.
Yeah, this is true. Speed O Pudding.
You are observant like me.
I am.
I'm
I
trying to
observe all.
That's Speed of Sound's Irish cousin.
Yep.
Speed O Pudding.
So I'm trying to find Christopher Sylvius's channel, but
I think Jerry posted it. He posted something.
did he?
The
Yeah,
side-
but
the side chat.
did he post a link to it? Oh
Yes.
no, he didn't.
Well, he posted a link to something. I can't click on it from my end. unless I go to the actual stream. It's on YouTube. But if I'm on StreamYard, I can't click on it.
Okay. I'm going to go check this out. Okay, let's see.
Exactly, Ox. That's exactly my point.
No, he posted a link to Anton Petrov. We just got closer to solving how life started on Earth.
I think that's clickbait.
Okay, truth be known, I started life on Earth. I'm actually really, really old.
You did it?
It
Explains
kind
a
of
lot,
seemed
doesn't
like
it?
your style there, girl.
It explains a lot, doesn't it?
My raccoons want to talk to you.
I'm like, ooh, animals. Let's just make them eat each other to survive. Ugh.
Got some complaints.
It's my perfect design.
I see Dessel is pointing me to something. What is he pointing to me at?
Let me go up, see if there's any more Dessel up there, or maybe it's the Anton link.
Yes, Gerby the Poppy. That's going to be the name of my upcoming stream whenever I stream next. Gerby the Poppy. The Poppy. The Pappy.
When are you going to stream next?
Oh, I don't know. I almost streamed yesterday, but then said,
I was waiting for you because I wanted to get basically I wanted to do something, and you fucking let me down again.
Yeah.
I'm really good at letting people down. I'm like a professional.
Speed's life is empty until that little red live label pops on, and then he perks up.
Well, as I say,
dogs.
As I say, I wish nothing but sorrow for you and your hearts. Because the only time you should be happy is when Gerstream starts. C.S. Lewis said that.
Normally,
how did I finish reading 1984?
Okay, I'm trying.
I actually think I ever read the whole thing. I listened to like an audio version of part of it, but no, I didn't finish it either way.
I read that when I was pretty young, really young, and it was. And I'm kind of sorry I did read it. Kind of depressed.
Because I mentioned the thought police. I see where that came from now.
Okay.
I read it in 1984.
Thank you, Randolph. I am full of rhymes.
Oh, did you really read it in 1984? That's cool.
Yep.
Now anyway, you can officially say I read 1984 and 1984.
Mm-hmm.
I'm gonna.
I can't say that because I was not born until after 1984.
You're just a whippersnapper.
I'm pretty sure
young punk.
I'm pretty sure a few minutes before I was born, there was a narrator that does commercials, and it said, "In a world gone mad, it just became a bit more mad. Germania has been born."
Now you can buy TV with DirecTV Auto.
Hey, boys and girls, we're done. I'm leaving.
Okay.
Hey, I'll tell you what, we'll take over. Me, Andrew, and Scott, we got this.
Yep.
Give me the access to this channel, full access.
Yep. Okay, so goodbye. Goodbye, cruel world. Anyway.